All right, we're back and we're going to look at cells of our innate immune system. And remember, innate is non-specific. So we went over and we talked about already some of the pieces of our innate system. Non-specific, something we just have. Remember, it's our skin. I drew this funky little person, showed you our skin on there. Tears, different things. So, but now we're going to get a little bit more specific about the cells. And you're going to learn how to identify the majority of these cells in hematology. So, we're going to start off with our granulocytes. Notice that has granules, okay, right there. And the other thing I'm going to point out is the three types all have, fill on them. So these guys are all filled with granules. Okay, so that's why filled with granules. Got it? Good. Okay, so our neutrophils, I'll draw a little picture. And I drew one of these in the other slide for you, and it just has little granules. Okay, our eosinophil, we're going to start out with a very similar cell. And then we're going to add orange granules all over it. Okay, and that's what an eosinophil looks like. And I have better pictures, but I thought, you guys, you like to watch me draw, right? Whoops. So our basophil looks similar, but this guy has really dark granules all over it. Okay, and they're black. And so that's what difference. And these granules all have different functions, of course. Okay, so let's keep going. Oh, what I want you to know is these granulocytes. Remember our CDs that we talked about? Okay, we have 45 on all of them, and then 15 is specific for granulocytes. Okay, and here's our neutrophils. Okay. This is the most abundant one. This is what you'll see the most of. So it's 50 to 70% of your white blood cells. You'll see it called a polymorphonuclear, okay, or you might see someone write a PMN. It's your PMNs, okay, and that's due to the shape here. You can see how that's drawn there. It's polymorphonuclear. Okay, this is the first cell to enter the site for an acute infection. And what I want, and I don't have it on here, these guys show up, okay? They show up when we have a bacterial infection. Okay? So that's what neutrophils, for bacterial infections, particularly. Okay? Right. They are active in phagocytosis, and if you remember what phagocytosis is, so these guys will come along, and I like to think of that as Pac-Man, phagocytizing and eating that, okay, digesting it. Some involvement in antigen presentation. So what that means is as it comes along, as it Pac-Mans it, it comes along, engulfs it. Then our next step, it's totally engulfed. Okay. We have it in there, and it starts to break it down then into little itty bitty pieces. Okay, and what it does is then it takes it and it presents part of that antigen, and it presents it to different things. A lot of times it's going to present it, okay, to a B cell, particularly if we're going to make some antibodies, and then we'll make antibodies to it so we can fight it off quicker next time. Okay. An eosinophil, and you can see those orange granules in here now. Pretty cool. And this is parasitic responses. So if you've got that nice tapeworm in your gut, okay, or you got that liver fluke that's managed to get in there, you're going to have an increased in your EOs. And that's pretty much where we see that. You'll see some of it as well in allergic reactions. One of the big things you're hearing about now is the eosinophilic asthma. And that's where individuals, usually due to an allergic response, and those eosinophils are just reproducing at a very high rate, and they're infiltrating the lungs. Okay, you don't see a lot of them. So basophils, this is the rarest. You don't see many of this guy at all, and you can see those little black dots in there. Unfortunately, a lot of times when he gets stained, those black dots actually get washed out, and then you don't see, he's not nearly that pretty. Again, we don't know 100% what all this guy's really good at. They may have a role in inflammation and allergies, and we definitely do know these granules get released, and they do have a bit of a histamine response. So we do know they a little bit bigger into the allergy response. But when we see a significant increase in these guys, we're going to think leukemia. Okay. We'll see some after patients undergoing radiation therapy too, a bit of an increase. Okay. But all of these guys work for phagocytizing, secreting those enzymes to destroy the organism. Okay. And then here's Mr. Monocyte. I call him really the garbage collector. Okay. He does a great job. So they're about 4 to 6% of what you're going to see normally floating around in your vascular system. What they do is they are involved in phagocytosis, and then they become that antigen-presenting cell. And a lot of times, you might see APC abbreviation, and that's what that means. Okay, and what these guys do is they change, and they go into the tissues and become those macrophages and dendritic cells. Okay. And they have the CD14 marker on them. And over here, what I'm going to point out, a lot of times that you're going to see in these monocytes are vacuoles. And in those vacuoles, what typically has happened is something was out here, it was phagocytized, was brought inside the cell. The enzymes broke it all down, and then you have this clearing, this vacuole is what's formed. So this guy's been busy, then engulfing things. And they don't just pick up organisms and pathogens, they just pick up dead cells. Okay. In an inflammatory process, we'll talk about in the next clip, there's just a lot of tissue damage that may occur. And these guys will just come in, and like I said, they're just the garbage collectors. They clean it all up for us. Okay. Now the lymphocyte, we cannot forget about the lymphocyte. He's really important in a lot of these things. And here's a lymphocyte right here. Okay. And we have our cytotoxic cells, or our CD8, okay, and our natural killer cells. And these two really have very similar functions. And sometimes in some of the videos, if you go out there and watch some, they're almost talking about them simultaneously. Okay, so unlike other lymphocytes, okay, almost all of our other lymphocytes are antigen specific. Okay, we don't want that big box. I just want a nice little circle around there. Okay, antigen specific. These ones are not. Okay, so our cytotoxic and our natural killer ones are not specific. Anything that invades, they're going to take care of. Okay, and what these guys are really good at are tumor cells, okay, and virally infected cells. So what happens with a tumor cell is you have it in your body, Okay? And this happens, I don't want to say all the time, daily, but it happens due to some sort of mutation in our bodies. We have this cell, okay, and something goes wrong with it when they're copying the RNA back to the DNA or the DNA to the RNA, and it gets messed up. And that's where cancer arises. And what these cytotoxic C cells are good at doing, okay, is recognizing that. And so these are our cancer cells right here. And what they're going to have on them, all of our cells, and this is specific to our cytotoxic T cells, and you'll see more with MHC complexes. They have MHC1 on them. And all of our nucleated cells have MHC1. So these cells, and they in here then are these abnormal proteins. And the cell knows, oh, something's not right. So it starts to display this protein on here, on its MHC1. And what happens? These T lymphocytes, they're doing their job, floating around, okay, and it has this detector on it. Okay, and it notices, oh, this guy has something on his MHC1 that's not normal. Okay, so it goes, it's attracted to it, okay, attaches to it, and this is where we have those perforins, the granulozymes, okay, and then this guy's also sending out other signals. Okay, and so it's saying, hey, I need some help over here, guys. We got something a little off the wall going on. We got to take care of this. So he calls in some more, and they start, okay, taking care of those tumor cells. So it takes care of them before they go anywhere else in the body, okay, and destroys them. And those perforins, I always think of it as a perforation. Okay? And when you perforate something, you put little holes in it. And that's what happens. It's putting these little holes in it, destroying the cell membrane. And we know what happens when the cell membrane is destroyed. The cell essentially lyses, disrupts it. Okay? And that's how those guys work. So we have our natural killer cells and our CD8 or cytotoxic T cells, and they can kill in two ways. So here's where I just drew you that nice little picture with that MHC1 complex. They bind to it, they secrete those perforins and those granulozymes to kill the cells. Okay. What natural killer cells can also bind to are antibodies on the coated cells. So you may have this cell, and we'll put some coronavirus in it because viruses need cells. Okay? And if they have this MHC on here, we do have some non-specific antibodies that'll say, uh-oh, that's weird. And so it's going to get coated with those. Oops, I made that one backwards. Having a little trouble there. Okay. And then these natural killer cells are going to find it and get rid of it. Okay. These guys are also triggered. Notice when I said we're sending out. Okay. This guy's sending out his own cytokines for help. But these cells as well, sick cells, so to speak, will give off some of this. And those are our cytokines. And another one is interleukin 2. That's something that you might come across. Interleukin 2. That's a big one for your natural killer cells. There are other interleukins. You can conclude that since this one's labeled 2, there must be a 1, and then there's probably more after that, and there is. Okay. Now these are all, okay, what's in your vascular system floating around. Now we can talk just about some of these cells that are specific to tissues. Okay? And those are our mast cells, our macrophages, and our dendritic cells. And so we'll look at some mast cells. And I did find some pictures these guys for you. You don't hardly ever see anything along this line. But our mast cell, it's similar to an appearance in a basophil. You can see it's got pretty granular on there. And again, its primary role is in allergic and parasitic infections. Okay? They have a surface receptor for that IgE. We'll talk about that a little more later, okay, when we get more into antibodies. Not as important right at this moment. Okay? But they contain granules of histamine and heparin. And what I want you to remember with these mast cells, when we talk about this histamine, when you are suffering from allergies, okay, what do you take? You take an antihistamine. Okay, so this guy's releasing these histamines, which is okay in certain situations. But when we have allergies, the problem with this is it's exploded. It's an overreaction. Okay, so it's found in the tissues, connective tissues, and near mucosal surfaces. So if you think about that, imagine it's your mast cells inside your nose. Okay, that's what I do. I think of them just, oh boy, that's quite a nose. Okay, so that's where your mast cells, those mucosal membranes. Okay, now we'll have our macrophages. The name of this guy is dependent on the location. So if it's in the blood, they're monocytes. When it's in tissue, it's a macrophage. Okay? So the image, what it looks like, is very similar. Okay? And I didn't find a great picture on the internet of macrophages. When I've seen them, when we've counted body fluids and that kind of stuff, and they look very similar to your mono with that nucleus. It's usually a fairly larger nucleus. And then the cell, this cytoplasm area is usually larger in a macrophage, and it usually has more vacuoles in it because this guy's busy at work, okay, engulfing things. And you can see in this picture down here, it's trying to depict that. You can see the vacuoles. It does appear to be a little larger, okay? And again, there's that CD14. We see an increase in number with inflammation, infection, and certain cancers. Not a lot of them, okay? They present those antigens. The reason why they can present those antigens is because they have phagocytized the pathogen, and then they broke it down, okay? So they engulfed it, and then they break it down into little pieces, and then they say, okay, here you go, B cells. I'm presenting you with this so you can remember it and make antibodies for next time. Okay. Here's our dendritic cells, and here's a nice little picture of them. Our dendritic cells have CD11. They're very active in phagocytosis and antigen presentation. And see, they're branched, okay? And so you can see that just makes, they get that bacteria, they bring it inside. You don't need to remember all these little chemical reactions. Here they call it they call this an autophagosome, okay? It engulfs it and puts it in this little thing. It secretes usually those enzymes and so forth in there to break it down. And then this is showing where it's presenting that antigen here. Okay. Let's see. Oh, this one's showing some fungi that we engulfed right here. Brought it inside with it. Okay. Over here, oh, I don't want you to pay too much to the excessive side over there. Okay. We're good right here. Let's put that back down. And oh, I did blow it up here for you, but we already looked at that. Okay. So the process, how exactly does our innate immune system work? We talked about the cells with it. And so what happens when we're exposed to something? And typically you have an inflammation originally, okay? And if you think about that, when you cut yourself, it usually does swell a little bit, okay? That's the inflammatory process occurs. Here's chemotaxis. I always look at chemotaxis, and it has the word taxi in it. And so what that does is that sends out those messages, and it mobilizes those cells, Okay, they need to mobilize and they need to get in that taxi and get to the site of where the inflammation is. They need to go where that angry coronavirus is. Okay, and then we have phagocytosis. Okay, I found this little Pac-Man guy. I always look at phagocytosis as Pac-Man. It's eating that. Okay. So what is inflammation? So inflammation is the reaction of your body to an injury or invasion. Okay? And here's an injury, hit my big toe, or did you cut your toenail too short and you've now got a bacterial infection in there. Either way, we got to do something with it. So imagine that invasion, okay, of that bacteria. Ooh, what could be one of those bacteria? Well, let's just go with Staph aureus right now. Okay. It brings a response to that infection. What do we need to fight that infection? We need our white blood cells. Then we need to eliminate it. All of our white blood cells showed up. It's eating this bacteria. And what happens to a cell once it usually eats it and destroys something on its inside? It usually dies too. Okay? So then our macrophages, our garbage collectors, need to show up and they need to take that away. Okay? And then we need to repair the damage. Okay? So inflammation. What happens with inflammation? Usually you have redness. Okay? Redness, swelling. Usually feels warm, doesn't it? It's usually painful. That big toe didn't look like it felt very good. Occasionally you might have some loss of function, depending on where the site is, what part of your body is involved. Okay? So the major events, you have an increased blood supply and capillary permeability. Why? This is where your swelling comes in. Is this important? Of course it is. Okay? If you have right here all of the blood cells showing up through your vascular system and the infection, is in your big toe out here, they've got to get to it. Okay? Because we don't want that infection getting into our vascular system because that's bad news. Okay? So we want that permeability so our cells can get there to fight it and keep it localized in this area. Okay? So that's where that migration of neutrophils and macrophages go. Okay? And then we have those chemical mediators that stop and start the response. So cytokines. Okay? And some of those are interleukins. I mentioned the interleukin 2 earlier. Okay? This is interferon. I don't know if I can spell this right. F-E-U-R-O-N. And then this one is tissue necrosis factor. Okay, so those are some of our chemical mediators. All right. So what do the chemotactic factors do, which we looked at? Okay. They attract cells to the site. And what are some of these? So complement pathways, one. We're going to look at the complement pathway, albeit briefly, but we are going to look at it. Our cytokines do that, okay, and other cellular products. So as this tissue damage is occurring, it is sending out these chemotactic factors to say, we got a problem. Houston, we have a problem, okay? And that brings the cells to phagocytize the invaders. That's how it works. We pretty much know what phagocytosis is. We talked about this before. Okay, it's the process where in the leukocyte engulfs and digests and kills the microbe. Okay, the cell attaches to the particle. You might see these PRRs and PAMPs, and they do talk about them in several of the videos. Those are just things that are on them. It says the peptido down here. You can see it, glycan in the cell wall. Okay. It recognizes those and binds it. You don't have to memorize that at this point. Okay, opsonins. Remember those opsonins? Okay, and what's crazy with immunology and everything that we talk about lately is we use so many words to describe such similar things. So here we have our complement again. Here's CRP, and our antibodies. So what these things do is I like it, the Greek word for it is to prepare food for. So if we have this guy and we put antibodies all over it, we're preparing it to be eaten. Okay? CRP is one. And remember what I call CRP? And I'm going to have a little thing about this too. Acute phase reactants. Okay, that's what CRP is. It's an acute phase reactant. So get it, it's acute, suddenly, phase reactant, and this really goes along with the inflammatory response. And if you remember, I say that CRP is elevated due to inflammation. And inflammation occurs for a variety of reasons. Okay? Now, what's interesting and gets tricky is some pathogens can survive in phagocytic cells and ride in these cells to spread infection. What's one thing that we learned in micro that helps with or what these bacteria use as a virulence factor against phagocytosis? Anyone remember before I say it? No, I know you can't answer me right now, but how about a capsule? Remember our capsule? Okay. And I just like this little picture down here where you can see, and I would say this looks like a dendritic cell. I'm not sure. I don't remember if this slide says what it is or not, but here's our microbe right here. And we notice that peptidoglycan in the cell wall, that PAMP, okay, it attached here. Okay, and we are engulfing it. We are phagocytizing it. And as it goes through here, you can see here's our vesicle. And when it's plain in that other cell and empty, we called it a vacuole. Okay. Here's our lysosomes, digestive enzymes. They migrate to there. They merge with this. Okay. And they start to digest it. They digest the microbe. Just like with us and our food, there's pieces of things that we just can't completely digest it, and we kind of poop it out, okay, or we present it, we could, as an antigen presenting cell, okay. And then here's just an electron micrograph picture of 1,2 phagocytizing the bacteria. Okay, all right, great job, guys. Thanks. I hope you didn't mind Schuster barking too much in the background.